Hello everyone and welcome back to another video. In today's episode, we're going to be discussing the eukaryotic cytoskeleton. So let's hop right into the video. So the cytoskeleton is an intricate network of protein filaments that extends throughout the cytoplasm. It's a filamentous architecture that is used to support the large volume of cytoplasm found in eukaryotic cells. And it's even more important in animal cells because they lack cell walls. So this gives it extra support through its dynamic structural nature. The cytoplasm of eukaryotic cells is supported and organized by this cytoplasm of intermediate filaments, microtubules, and actin filaments, also called microfilaments. So let's begin with microtubules. Microtubules are stiff, hollow tubes formed by globular tubulin dimers. This dimer is a heterodimer, hetero meaning different subunits. The opposite version of hetero would be homo, and dimer meaning we're talking about two different subunits here. So these globular tubulin dimers form this giant polarized structure with a slow-growing minus end and a fast-growing plus end, as displayed here. These microtubules display dynamic instability, which refers to the alternating nature between growth and shrinkage. And this is a rapid process. This process is catalyzed using GTP. So shrinkage is promoted by the hydrolysis of the GTP that's tightly bound to the tubulin dimers, reducing the affinity of the dimers for their neighbors and thereby promoting microtubule disassembly. We've heard of microtubules before. In meiosis and mitosis, they grow out of the organelle known as the centrosome, in which the minus end remains embedded within the centrosome, and the plus ends grow outwards. Here's a microscopic display of a microtubule. We see the dimers, and there's 13 of these columns, you may think of them as, and they're known as protofilaments. So what should you take away from microtubules? Well, let's list their functions real quickly. Microtubules are highly important in organelle and vesicle transport, nuclear division, mitosis, and cell motility. Another important feature of microtubules is seen here. Kinesins and dyneins are microtubule-associated motor proteins that use the energy of ATP hydrolysis to move unidirectionally along microtubules. These proteins carry specific organelles, vesicles, and other types of cargo to particular regions of the cell. So now let's move on to intermediate filaments. Intermediate filaments are stable rope-like polymers built from fibrous protein subunits that give mechanical strength to the cell. That is the most important part of intermediate filament function. It provides that tensile strength. Cytoplasmic intermediate filaments are as follows: the keratin filaments, vimentin filaments, and neurofilaments. And lastly, nuclear intermediate filaments we see in the nuclear lamina, which stabilizes the nucleus. So when we say intermediate filaments provide good tensile strength for the cell, what we mean by that is it provides the cell with an ability to withstand tension without breaking. Think of like strands of a cable. From what we know about protein structure, we know that this must be alpha helices. Must be. The coiled nature of intermediate filament monomers and dimers and moving into the tetramers, just the congregation of the structure, we can deduce that information just from this. So following multiple anti-parallel staggerings of these monomers and then eventually the dimers, we get to the tetramer. This continues on and you get this giant lateral association of the tetramers. And this is where we see the sheer tensile strength displayed by this structure. And one last thing with intermediate filaments to keep in mind is that they are highly involved in cell-to-cell junctions, as we see here, along the lengths of each of these cells. And lastly, microfilaments or actin filaments. These actin filaments are helical polymers of globular actin monomers. Actin filaments are the most flexible of the cytoskeletal elements, and they're usually found in bundles or networks. Just like microtubules, these microfilaments or actin filaments are polarized with a fast-growing plus end and a slow-growing minus end. And their assembly and disassembly is controlled by the hydrolysis of ATP. Unlike microtubules, which is GTP, this is ATP. The ATP is tightly bound to each of the monomers, and it's controlled by various actin-binding proteins. So what do we see microfilaments functioning in? Well, they provide strength and structure for the cell cortex. So what do we see microfilaments functioning in? Well, they provide structure for the cell cortex. They're also highly prominent in muscle contraction, which we see in sarcomeres. I forgot to mention, with regards to the cell cortex, there's a concentrated network of these actin filaments underneath the plasma membrane, and that's what the cell cortex is. It's responsible for shape and the movement of cell surface, including pseudopod formation, which occurs through cytoplasmic streaming. This is something you'll witness in the lab setting. And lastly, cytokinesis. The microtubules function in cytokinesis by pinching the cell through that cleavage furrow, which we can see displayed here. So two cells pinching following telophase, and there's a contractile ring of actin and myosin filaments within the cleavage furrow. And here's a display of what the dynamic instability of the actin filament looks like, with the minus end and the plus end, and the hydrolysis of ATP. So under each of these, let's write a couple notes so you can take this away with you. Microtubules display dynamic instability. So does actin filaments. Intermediate filaments do not display dynamic instability. Well, how about functions? Let's name a few for microtubules. Cell motility, spindle fibers in meiotic and mitotic division, and intracellular transport, meaning the movement of vesicles and organelles within the cytoplasm. So intermediate filaments, tensile strength. Think of keratin, think of the nuclear lamina. And actin filaments, think of sarcomeres found within muscle contraction function. And think of the cleavage furrow. The pinching of the cell following telophase. So I hope this video cleared up some of the basics of cytoskeletal function, what elements compose a cytoskeleton, and the functions that each of them display. In the near future, I'll probably go into each of these individually and make a more in-depth video for microtubules, for intermediate filaments, and for actin filaments. But until then, utilize this video to differentiate between the three. So I hope you enjoyed. We'll see you in the next one.